Hi, sir. You can actually start the session right
Yeah. Hello, dear students. A warm welcome to the revision classes on important topics. I know you are on the verge of your university examinations and almost may have covered almost all the topics. But even then, just I'm trying to remind you these are the important topics. Whether you have missed it, we can just go through that. We'll just anyway have a look at each and every topics one by one. So there are only two options for you, either make progress or make excuses. Even now you have time to progress. That's what I would like to remind you. And let me straight away start each topic by topic. First, the topic what you already, the first you studied in your classes, enzymes. Enzyme, at least once in 3 units examination, either an essay or short essay comes from that topic. So it's very important topic, I should say. And of course, the two essay questions that you already asked, one is always about the factors that affect enzyme activity. As you know, you are supposed to say that each topic by point. In the factors, you're supposed to draw the diagrams to show the how the pH affects, how the enzyme concentration, substrate concentration, and product concentration, how the pH affects the enzyme activity. So these all should be explained with the help of a diagram. And usually for the examination, when the essay comes, it comes along with the classification of enzymes. Classification of enzymes, I should remind you one thing, we always write in the order, because whenever we say enzyme class 1, it is oxidoreductase. Enzyme class 2 is transferase. Enzyme class 3 is hydrolase, like that. So that you should remember. And then, as you know, now in the new books, we have already added one more. Earlier, we had only 6 classes. Now we have a 7th class called as translocase. So don't forget to write about that also. As a 7th class of enzyme is translocase. So please add that also. And next question, next another question that can be asked in essay is again a combination of isoenzymes and clinically important enzymes. It can be either they can ask you related to medical myocardial infarctions or related to liver disease. So these are the two essays we expect from this topic. And of course, as a short essay, in general, the examiners can ask you on enzyme inhibition, where you're supposed to write about competitive, non-competitive, uncompetitive, suicide inhibition, and allosteric inhibitions. Of course. Okay. Then the short notes on enzymes. So number of times the question asked is about the fourth one, competitive inhibition. That's the most important question, I should say. Whenever you write about the competitive inhibition, not only the features, you should say the clinical importance of it. What's the clinical importance? Most of the pharmacological action of drugs is by competitive inhibition. That's an important question. Then Km value has been asked. Now I have seen this as a separate question, either the role of substrate concentration on enzyme activity or the role of pH on enzyme activity, like that. That also can be asked in 3-mark question, the short question. And Km value and natural enzyme specificity. Enzyme specificity, there's another question where we say about absolute specific, group specific, bond specific, stereo specific. So that should be added up. You should also go through that topic too. So these are the major questions from the enzymes. Next is on vitamins. Vitamins, usually the essay question that has been asked are either from vitamin A, vitamin D, or vitamin B1. And always the question is a direct question where it's asked about write the chemistry, sources, RDA, biochemical functions, and deficiency manifestations of vitamin A or vitamin D or vitamin B1 can be asked like that. So very repeatedly asked question is vitamin D, as you know. Then about in the short essays, I have given you the functions and deficiency manifestations of vitamin C. Always, that's also another important repeated question. once or twice this question, functions and deficiency manifestations of B12 has been asked. And in short answers, short questions, natural antioxidants. I hope you know about it, that natural antioxidant means that the question is on vitamin E. Yeah, so that can be one question. Then vitamin K cycle can be another question. Then some clinical manifestations, beriberi, pellagra can be asked as a question. Beriberi has been asked several times. Then in general, once the question about niacin, and once just a straight question, PLP, pyridoxal phosphate, about the B6 functions of B6. That's what we expect from you. That has been asked. And once again, in short answer, TPP has been asked. Since that was given there, I didn't repeat it here. So TPP or thymine pyrophosphate, that's also another short answer question. In this anyway, A and D has been asked several times. Once or twice B1 has been asked, and scurvy or vitamin C is another repeated questions of vitamin from the part of vitamins. Now from the vitamin, I'm just going to minerals, because whenever you study about the vitamin D, you should study on, or along with that, study about the calcium also. A normal question, I always repeat question, normal level of blood calcium and how is it maintained? What are the factors in maintaining blood calcium level? I would like to say why I put minerals here, just because, as you know, the major function of vitamin D is to maintain the blood calcium level. So one of the factor that maintain blood calcium level is vitamin D. It's active form calcitriol. So that's why I put together. So whenever vitamin D study along with that, you study about the blood calcium also. And here, one important thing you should remember, whenever a question is about the normal level of blood calcium, it is not just the value that is not the one what we expect from you. We know the blood calcium level, generally we say 9 to 11 milligrams per deciliter. That alone is not the one we expect from you. The thing is, in the blood, calcium exists in 3 forms. One is ionized form, another one is complex form, and protein-bound form. In that, ionized form, which is around 50%, is metabolically active. So, as a student, you should know that the ionized calcium is the one which is metabolically active. So, you are supposed to say about that. After writing the normal value, say it exists in 3 forms in the blood. In that, 50% is ionized calcium, and that is metabolically active. So that much we expect from you when we ask about the normal value of blood calcium level. Then how is it maintained? Then you are supposed to say about 2 things, hormonal and non-hormonal. Hormonal factors, of course, you know, vitamin D's active form calcitriol act like a hormone. So calcitriol, PTH, and calcitonin are the hormonal factors. And there are some non-hormonal factors like the blood phosphorus level, age, children, renal threshold. These all should come in the and blood protein level, acidosis or alkalosis. So that are the non-hormonal factors. So just in the beginning, is it an essay question? If it comes as an essay question, just divide it to, just we can put as like a synopsis even, and put it into hormonal and non-hormonal factors, then write, mention the names and explain. This is what we expect from you. And another question that is always asked is about the iron absorption. The question can be either iron absorption or mucosal block theory. If the question is on mucosal block theory, you are supposed to say about iron absorption also. And if the question is just iron absorption, you are supposed to say about mucosal block theory, because that's a theory which explains how the iron absorption is maintained. Why iron absorption is important? Because that's the one, the level of iron is absorbed, the level of iron in the blood is maintained at the level of absorption. I repeat, the level of iron in the blood is maintained, or the body is maintained at the level of absorption. How? We are explained through a mucosal block theory. So these should be written together. That's another short essay, or sometimes it comes as a 3-mark question. Then another short answer question always repeatedly asked is fluorosis. That's why I put in bold letter. Fluorosis, or sometimes a separate question I have seen, functions of fluoride also. And then one more question that what we can expect is about the copper. You just study the function of copper. Along with that, you study about this word Wilson disease. This is about iron accumulation. Okay, Wilson disease is because of copper accumulation in the body, because from the liver it is not excreted out. Okay, that's what it's all about. So Wilson disease, that also is studied. Next topic is about the carbohydrate metabolism. Carbohydrate metabolism, 4 major questions so far has been asked in essays once or twice. One is about the glycolysis, number of times gluconeogenesis, and gluconeogenesis straight away ask a question of gluconeogenesis, or sometimes how glucose is synthesized in the body. Then you should say the non-carbohydrate sources and say about the sources one by one, and list what are the sources of glucose synthesis, and then how the process regulated, major steps and regulation. And third question is about glycogen metabolism. A regulation of glycogen metabolism alone has been asked 1 or 2 times. So study that too. Then blood sugar regulation. That has been asked in several ways. How the blood sugar is maintained in fasting conditions or in post-meal condition after food, or explain about the hormonal regulation about blood sugar level. What is the significance of it? These in different ways the question has been asked. So study about that. That's an important question anyway. So blood sugar regulation, when it is asked, right, if it's a general question, write about the hormonal factors, then the role of organs that maintain blood sugar level. That's also important. Write about that, study about that too. And mainly about liver. When we say about the organ that maintain blood sugar level, we should say about the liver, should say about the pancreas, muscle, adipose tissue. Okay, short answer questions. From the chemistry part, the major question is mucopolysaccharide, or another word, glycosaminoglycans, because that's a major compound present in any extracellular matrix. So in that way, it's important. Or sometimes the question can be twisting one or risky one. They can be asked as heteropolysaccharide, and there you can say about other heteropolysaccharides also. Anyway, don't forget to write about glycosaminoglycans or mucopolysaccharide if the question is about heteropolysaccharide. Another question, we think it is a very simpler one, is about the digestion absorption of carbohydrate. I've seen simply the students now simply they will write about it in some ideas about that, but the most important thing is when we say about the digestion, when we say about each enzyme action, how it acts, that is important. So say about, for example, salivary amylase action, you should say it randomly breaks alpha-1,4 glycosidic linkage. It mainly is the site of action is mouth, and action is randomly cuts alpha-1,4 glycosidic linkage. So in that way, make specific answers. When you write about each enzyme action, be specific in your answer. That's what we expect from you. Then another question, repeated question, is about the significance of HMB pathway. In that, as you know, glucose metabolism, most of the pathways for the glucose is the breakdown of glucose, thereby we get energy. But this pathway is not for energy. Then for what? That's what we expect from you. Then for what? 2 major compounds are produced. Specialized compounds are produced from HMB pathway. They are, one is NADPH plus H plus, and another one is pentose phosphate. So you're supposed to say about that. Supposed to say the functions of that. When you ask about significance of HMB pathway, say it's for to synthesize specialized compounds, namely NADPH plus H plus and pentose phosphate, then its functions. Another question can be about galactosemia. And another question is dietary fiber. A number of times it has been asked. Why I put the dietary fiber along with carbohydrate? As you know, I think you know that dietary fibers are carbohydrate, indigestible carbohydrate, like our starch. I'm sorry, not starch. Our cellulose, pectin, lectin. So these are the hemicellulose. So these ideas you should have in your mind when you say about dietary fiber. So dietary fibers are indigestible carbohydrate. That basic concept you should know. And the 6th question what I've given here is von Wieker's disease. which is nothing but a disorder related to glycogen metabolism. So it's about the glucose 6-phosphatase deficiency. So that also can be asked in 3-mark question. Next topic is about the lipid metabolism. Many times the question from the lipid metabolism that has been asked is about the beta oxidation. We have already explained that in detail in the last our class, revision class. Beta oxidation can be an essay. I taught you how to write. So whenever you write any answer, please write it in order with subheadings. I gave one example about beta oxidations on that day. Okay, so that's one question. Second question can be on ketone bodies, synthesis, utilization, and about importance of ketoacidosis. A number of times, ketoacidosis or ketosis has been asked as a short essay question or short answer question. Lipoprotein in general, once along with the, please note, along once it has been asked in this way, classification of enzyme and then write on lipoproteins. Once it was an essay. The cholesterol can be asked as a part of synthesis, or mainly the fate of cholesterol is a major question from that. Then short answer questions. Of course, one repeated question in the last 10 years is one essential fatty acid. And whenever the students making mistakes here is that whenever the essential fatty acid is asked, some students will write about essential amino acid. When essential amino acid is asked, some student by mistake, they write about essential fatty acid. And remember, fatty acid is asked, just think FA is there. You think in this way, FALL, FALL. That L is for linoleic acid, linolenic acid, and A for arachidonic acid. So essential fatty acid, just with one word, FALL, FALL. You can just remember the things. Don't make mistake in that. And not only the names, what are its function, what are its deficiency manifestation. That's what we meant by that. So write that, say the definition, then name, then functions, deficiency manifestation. If you are able to write about the sources, fine. Another question from the chemistry of lipids is phospholipid. Once or twice it has been asked in this way also, name 4 phospholipid and mention their functions. Phospholipid, as you know, it's an amphipathic nature, it's a part of your cell membrane, it's a part of your lipoprotein, it's a part of your micelle formations. So in that way, just we can understand that because of its amphipathic nature. So glycophospholipid, classify it, glycophospholipid and sphingophospholipid, write about each name and its function. Like we studied in digestion and absorption of carbohydrate, the same way, digestion and absorption of lipid is another question. So in here also, just say what happens in the mouth, what happens in intestine, in stomach, what happens in intestine, and how is it absorbed. Don't forget to say about our micelle formations and also about when you say about the digestion, write about the emulsification. And based on this, one question that can be you should study is about bile salts. Bile salts is one of the fate of cholesterol, you know it. Okay, so when the question is asked about bile salts, say about its synthesis from the cholesterol and then about its function. One is in the process of emulsification, another function is in the process of micelle formation. So by emulsification helps in the digestion of lipids, of fat, and by micelle formation help in the absorption of fat. So study that. Another question is about HDL metabolism. Is another question we have already discussed in our last revision class. So I'm not going to say about that. We'll go for the next topic. Is about the protein. So carbohydrate over lipid protein metabolism. In that, a repeated question that has been asked is on tyrosine metabolism. Either tyrosine metabolism as such, I'll show you another question later. Tyrosine metabolism as such and what are the compounds, specialized products. It can be a short essay question as such itself. That's one question. Another question that can be aspect is urea cycle. Either the question can be urea cycle or it can be asked, what are the sources of ammonia and how ammonia is disposed from the body. So by forming urea, since ammonia is, so this word you should remember, ammonia is neurotoxic. So the body converts ammonia into urea. So whenever the urea question is asked, these all matters. What is urea? Where is synthesized? And Why is it synthesized? Urea, because to detoxify ammonia. So that question can ask in that way also, how ammonia is disposed from the body. Thereby, indirectly, the examiner is asking you on urea cycle. Another question is on glycine metabolism, mainly what are the biochemically important compounds produced from glycine? There you can have two questions. One is biochemically important compounds produced from glycine or biological importance of glycine. If the question is biochemically important compounds produced from glycine, you can straight away go for glutathione, purine, creatinine, collagen, heme. But if the question is about what are the biological functions of glycine, not only this synthesis part of specialized products, but also it helps in the conjugation of bile acid, conjugation of benzoic acid and its excretion, act as a neurotransmitter. That also should be added up. Please not. So please understand the question and write whether just question is about biological importance or biologically important compounds from glycine. The short answer questions can be either again essential amino acid. They're essential fatty acid lipids, here essential amino acids. We have 8 essential amino acids and 2 semi-essential amino acids, and say about its function, say about its, what will you explain other than that? In 3 mark question, just by writing 3 names by 10 words, you won't get the 3 marks. Then what more you're supposed to write? You should say about complete protein, incomplete, and poor protein when the question is about essential amino acid. Any protein that which contain all the essential amino acids called as complete protein, or milk protein, or egg albumin is a complete protein. Then incomplete protein, which lacks 1 essential amino acid, and poor protein, which lacks more than 1 essential amino acid. So add that also. Then only we can give you all the 3 marks for that. Another question is structure of protein, either the primary structure or secondary structure. And I'll tell you long back, around 10 years before, this structure of protein itself has been asked as an essay. Explain the various levels of structure of protein with example was the question. So anyway, now last 10 years, once or twice it has been asked primary, or once or twice the secondary structure of protein has been asked. Okay. Another question is transamination. I will say about that later, because that question can be used in several places. Another one is transmethylation. Then you are supposed to say about the active form of methionine. It is S-adenosyl methionine. So whenever the transmethylation is asked, say about SAM, S-adenosyl methionine, its synthesis, then about transmethylation reaction, because that's the one which helps in transmethylation. Then 2 disorders related to tyrosine metabolism and phenylalanine metabolism, that's phenylketonuria and alkaptonuria, can be a short notes. And around 3 or 4 times, this question has been asked. PEM. What is PEM? Protein, energy, malnutrition. Kwashiorkor and marasmus. That also it should go through. It's also an important question. Now, some topics about transamination. Why I put here transamination reaction? Just because this you can answer in number of places. One, if the question is about enzyme classification, the class 2 is transferase. These 2 you should write an example. Alanine plus alpha ketoglutarate giving pyruvate plus glutamic acid, or aspartate plus alpha ketoglutarate giving oxaloacetate plus glutamate. So there's an example. So that's one question where you can utilize this. Another question can be this straight away transamination, transfer of an alpha amino group from an alpha amino acid to an alpha keto acid to form a new alpha amino acid and new keto acid. That's another one. There, transamination reaction. Yeah. Here, what's up? Then after writing definition example, what will you write? Here you are supposed to write about the significance. What are the major significance of transamination reaction? Here, you have seen all the 4 words there. Alanine, alpha ketoglutarate, pyruvate, glutamate. Same way, aspartate, alpha ketoglutarate, oxaloacetate, glutamate. So here, just think about each and answer it. First, you saw the word amino acid alanine. So your first importance of clinical importance of or significance of transamination is, that's the first step. Transamination is the first step in the catabolism of any amino acid. That's your first point. Second point, alpha ketoglutarate. In most of the reactions, alpha ketoglutarate is the alpha keto acid which accept the amino group because it can undergo deamination at a faster rate. So second point. Third here, pyruvate or oxalate. What's the importance of that? They are the sources for gluconeogenesis. So transamination process provides substrate for gluconeogenesis during the fasting conditions. So that's the third significance of transamination. Last one, we have seen glutamate. That means what? Most of the non-essential amino acids can be synthesized by transamination. So from each word, you get 1 point. And then one more, another place where we can use this reaction is about the liver function test. As you know, AST and ALT are the enzymes that are elevates in hepatocellular diseases. So here for transamination, we can write as a clinical significance. Other than that, liver function test, it can be written AST, ALT estimation is one of the major tests what we use to know the liver function because that will get elevated in hepatocellular diseases. In that, ALT will be elevated very high during the acute liver diseases. That also you should supposed to know. So third place where you can write about this, where we are supposed to know is in liver function test. Fourth topic where you can study this is, as I said earlier, gluconeogenesis. One of the major sources of gluconeogenesis is amino acid, glucogenic amino acid. The compounds produced by transamination like pyruvate, oxalate. Okay, these can be used or can be used as source for gluconeogenesis. So that's why I put this here. So transamination reaction, when you study, it has about, see, we can use this in 4 other different topics too in biochemistry itself. Okay, then I'll just show some questions in that has been asked as essay in protein. See, list the different sources of ammonia and explain how ammonia is transported. Describe the reactions of ammonia detoxification in liver. Mention the uses of increased blood urea level. See, once this was essay. See, from here, in the third sentence, describe the reactions of ammonia detoxification. That is about the urea side. And last is about what's the normal blood urea level and what is important. Blood urea level is elevated in renal disorder, renal dysfunction. And first one, what are the different sources? There, one is amino acid is a source. From amino acid itself, in 3 ways, the ammonia is produced. One is a combination of transamination and oxidative deamination. Together we call it as transdeamination. Another source is non-oxidative deamination. Another source of ammonia from amino acid is action of L-amino acid oxidase. So 3 different sources for ammonia. And so, from the amino acid itself. Other sources are purine pyrimidine degradation is another source of ammonia. Another source is action of intestinal bacteria on the food. So we have 5 different sources for ammonia. Please note. And explain how ammonia is transported. From the brain, it is transported as glutamine, and from the muscles, it is transported as alanine. We have studied that as you are studying that as a part of gluconeogenesis, glucose alanine cycle or Cargill cycle. So remember that also. So this way the question can be asked. Another, once the question has been asked in this way, define transmethylation, discuss about the synthesis, functions, and catabolism of amino acid involved in transmethylation reaction. It's all about methionine and cysteine. Study that. And disorder is homocystinuria. When you study homocystinuria, you'll get answer for the folate trap from the B12. Deficiency manifestation of B12, folate trap. You'll get a number of questions can be asked that there also. So number of things are involved in that. So study that. Next question is about aromatic amino acid. And the question came in this way. Name aromatic amino acids. The word phenylalanine, tyrosine, tryptophan are aromatic amino acid. Discuss the metabolism of any one of them. So they're indirectly asking to write about tyrosine metabolism. And add a note on associated metabolic disorders, meaning the question on phenylketonuria, alkaptonuria, and albinism. So here, any amino acid's name has not been mentioned, but the question is about, as you know, it can be on phenylalanine and tyrosine metabolism. So these are the ways in which the question can be asked on the protein metabolisms. And once there's a separate question like serum electrophoresis, when we do at the plasma protein, mainly the question is about the plasma protein electrophoresis. What happened? How many bands you get? You know about albumin band, alpha 1, alpha 2, beta, and gamma band. 5 bands you get when you do the serum electrophoresis. So that's a technique what we use to differentiate or separate the biomolecules based on the charges, mainly based on the charges and the size of the biomolecule. And plasma protein can be, or serum protein can be separated into 5 different bands by serum electrophoresis. Okay. Another topic is hemoglobin heme metabolism. Here, 2 repeated questions are there. One is about the heme synthesis and regulation. Another one, heme catabolism and jaundice. So this is one important topic. Anywhere, somewhere, either in your theory or in practical examination, number of times we ask this question, because in abnormal urine analysis, when we say we will be giving you the urine sample of an obstructive jaundice case. So either in theory and practical, this is very important. So study this topic. And in this question, the question can be in a crispy one, differential diagnosis of jaundice, or otherwise classify jaundice and mention the laboratory findings of each category. Or other words, what are the changes occurs for the biochemical parameters present in blood and urine in different jaundice conditions. These all are the same. Differential diagnosis of jaundice. There you're supposed to say about the biochemical parameters in the blood, mainly bilirubin, in that total, direct and indirect. Then AST, ALT, ALP, 3 enzymes. These are the 6 biochemical parameters in the blood that get altered during jaundice. Then in the urine, it can be on the bile salt and bile pigment. Feces again, stercobilin. So this is what you're supposed to write in differential diagnosis of jaundice. Write the names of the parameters, write the normal value, what is elevated in which condition. This is what you're supposed to know. That is an important question anyway, in both theory and practicals. Same question, normal, what's the normal serum level and formation of bilirubin? Is nothing but the heme catabolism. Short answer question. Once the question was on hemoglobin structure and function. That means contain 4 globin chains and heme. That's what about the structure you're supposed to say about here. And you can say if you know about the primary and secondary structure of that, you can add about that structure also. Functions about hemoglobin. We all know about the transport of oxygen or carbon dioxide. Not only that, it acts as a buffer. That you should know anyway. It acts as a buffer. And it has a major role in maintaining the pH of the blood. So you are supposed to write about that. Okay. Then short questions. Once the Bohr effect has been asked. Once or twice sickle cell hemoglobin asked. Once or twice thalassemia has been asked. And along with the heme synthesis, you are supposed to know the disorders related to it. There, you are supposed to say about porphyria. And once or twice AIP, acute intermittent porphyria, has been asked separately as a short answer question. Okay, another topic is about nucleic acid metabolism and molecular biology. Here we have not a regular one, but a question is there. What is uric acid? How is it synthesized? When you ask me how the uric acid is synthesized, meaning about the purine degradation, okay, and state the reason for hypo or hyperuricemia. That means you're asking about, the question is about the reasons for gout, the reason for the primary gout and secondary gout. So the reasons can be added up. Then short questions, biologically important nucleotides can be a short question, gout can be a short question, structure of DNA, structure of about the transfer RNA, tRNA can be a question, codons or genetic code and its features can be another short note. Antibiotics interfering in transcription has been asked once. Means what? What are the inhibitors of transcription? I'll show you some other surprise question later. So these are some questions that can be asked in nucleic acid metabolism and in molecular biology. Then about acid-base balance. We have a repeated question here about the physiological pH of the blood. Mention the mechanisms and explain in detail about one. And in that, so many a times, this question, renal regulation of physiological pH, we know about the 4 chart-like, 4 tabular columns. That's another important question. Then short answer question about blood buffers, alkali. Another question, what is alkali reserve and its importance? And number of times metabolic acidosis can be asked. Next question is about the function test. Liver, as I said earlier, liver function test can be a question. Once it was been asked as an essay, liver function test and its significance has been asked. There, we're supposed to say about total protein, albumin, bilirubin, direct, indirect. Then SGOT, SGPT, or AST, ALT, and ALP. That's it. Write about these things. In what are the normal value of these 6 and in which all condition it increase or decrease. Please add up that. That's an important question. Then about the kidney function test, once it has been asked in general in a short essay question, and clearance test, or specifically creatine clearance test, has been asked several times. Then regarding the biological oxidation, we have already studied in detail in last revision classes. Definition of oxidative phosphorylation, components of electron transport chain, chemiosmotic theory, high energy compound, uncoupler. In that, you can expect one question. And after that, these are the major topics from which the question can be asked. And as I told you, every year, some surprise question I have seen. I've just put it together. Once the surprise question was epimerism. Epimer. So epimer anomer can be a separate question. Benedict test, anyway, you are doing the practical classes, so that's another question. Homopolysaccharide, not hetero, homopolysaccharide. Our starch, glucagon, agar, starch, glucagon, then inulin, fructose, a polymer fructose. These all comes in homopolysaccharide. That can be asked. BMR was a short nod once. Nitrogen balance, another question has been asked. Then functions of fluoride. Fluorosis is a question we can understand. Functions of fluoride have been asked once. As I told you earlier, another separate question was serum electrophoresis. Once a separate question was ELISA. Again, this all comes in 3 marks. Another question, Beer-Lambert's law in colorimeter. Then inhibitors of replication, transcription, translation. And once the question I didn't write here, write an outline on the steps of DNA replication was another some separate question. Anyway, so I'm trying to cover almost all the questions that have been asked in last around 15 years in our university examinations. So I hope you'll be able to go through, and whenever you write the answers, as I told you earlier, as I taught you earlier, try to write with subheadings. Wherever normal values comes, you put the normal values. Wherever you can write schematically, you can arrange the things in the order. Then also that also is important. You can add up that. Put it in such a way that there should be some clarity. The examiner should be able to get an impression about you. In that way, write the answers in that way. Have some clarity in that. Don't scribble, don't just simply put everything together and in such a way that nobody can read it. When you put that, an examiner can easily understand, okay, that fellow has not studied, simply is trying to put something just for the sake of wasting the time and the paper. So don't do that. It will give a bad impression. Anyway, whenever you study anything, study in this way with subheadings. And I hope you all can do well in the next examination. Please go through these questions and all the best. Thank you for the patient listening.